Continuing on in chapter 22, looking more in depth into the heart. So the little bit here about some histology of the heart. So whenever we start looking at this, back to our cardiac muscle that we talked about in A&P1, that cardiac muscle is striated, it is branched, that muscle tissue. Again, we have the connective tissue on the outside. We have the sarcolemma, which is the plasma membrane of those muscle cells. The mitochondria, of course, lots of those for that contraction. The myofibrils, which are the individual muscle fibers that run through here. They have the sarcomere, which is the individual contractile unit. We have these intercalated discs that allow for exchange of communication and nutrients, as well as calcium between cardiac muscle cells. Again, these are branching muscle cells that we see that differentiates them from some of the other muscle cells. And then our sarcoplasmic reticulum, where we store some of that calcium in there. So we look again at these cardiac muscles. They're elongated, branching, 1 to 2 about centrally located nuclei. We have the actin and myosin myofilaments. It is striated, where we get those sarcomeres. The sarcoplasmic reticulum, again, stores the calcium, but not in dilated cisterna that we see in the skeletal muscle. They have those intercalated discs that I'd mentioned that allow that cell-to-cell contact. We have desmosomes to hold the cells together. And then there are also gap junctions that allow for action potentials to move from one cell to the next. So with all that connection, electrically speaking, cardiac muscles between the atria and the ventricles essentially behave as a single unit. So this conducting system relays the action potentials. So it's modified cardiac muscle cells that form 2 nodes in a conducting bundle. So the 2 nodes to start with, the SA node, which is the sinoatrial node. This is called the pacemaker, and it's a specialized cardiac muscle cell, and it generates spontaneous action potentials that pass to the atrial muscle cells and to the AV node. That AV node is the atrioventricular node, and this is where action potentials are conducted much more slowly, and that ensures that the ventricles receive the signal to contract after the atria have contracted, because you wouldn't want ventricles contracting before the atria, so the ventricular ones, that action potential slows down to ensure the atria contracts first. The AV bundle passes through a hole in the fibrous skeleton to reach the interventricular septum. Then we also have the right and left bundle branches that extend below that endocardium to the apex. And finally, the Purkinje fibers. This is a large diameter cardiac muscle cells with few myofibrils, but have lots of gap junctions to allow conduction of the action potential to the ventricular muscle cells. So we look at this conducting heart. So the heart is at rest. All the chambers here are relaxed. Then as the action potentials are generated in that SA node, they spread from that node to the adjacent cardiac cells of the atrium. The preferential pathway conduct those action potentials that we see here in the dotted lines. So that's going to move those from the SA node to the AV node with greater velocity, so much faster than they're transmitted to the remainder of the atrial muscle cells. However, such pathways cannot be distinguished structurally from other areas in the atrial wall. Okay, then the atria contract. So the cardiac muscles of the atrial wall contract in response to that action potential enough once they're conducted through that atrial wall. So now we have the contraction of the atria, which is shown here by these little wobbly lines here. So we get the contraction of the right and left atria. Then the action potential go more slowly through that AV node. And remember, that's to allow the atria to contract first. So they first come into that SA node. Then they move quickly through the atria, but slower through the ventricles. So due in part to the smaller diameter there of the muscle cells, again, also fewer gap junctions in the intercalated disc. So that's going to help slow down the spread of that action potential. Again, that delay at the AV node allows for completion, so the atria contract before the ventricles contract. Now the ventricles can contract. So because of the arrangement of that conducting system, the first part of the ventricular myocardium that's stimulated is the inner wall near the apex. So again, we kind of see down here closer to the bottom. So again, due to that kind of slow moving through that interventricular septum, we actually get the apex contracting first. So it begins there and it progresses through toward the base of the heart. So kind of a spiral arrangement of the muscle layers results in kind of a ringing action of that contraction. All right, now if we look at the electrical properties, so like skeletal muscle, the cardiac have a normal resting membrane potential, and they can conduct action potentials. So that resting membrane potential depends on low permeability of sodium and calcium, but more permeable to potassium. So there's potassium leak channels, like we talked about with neurons. Potassium is the main thing moving at rest. Now we do have some autorhythmicity. So the heart is autorhythmic, meaning it stimulates itself to contract at regular intervals. And again, that's the pacemaker cells in the SA node that can generate those action potentials spontaneously. So that pacemaker potential, it spontaneously developing local potential. It's due to the movement of sodium, potassium, and calcium. So once we get to that threshold, then an action potential would be generated. So if we look at this pacemaker potential, so kind of here at rest, this pacemaker potential, sodium ions cause depolarization. That's similar to what we talked about with neurons through specialized non-gated sodium channels. So that decreases the permeability of potassium. It no longer wants to go out, and that further decreases or causes more of a depolarization as less potassium comes out. So sodium's moving in, potassium's kind of trapped inside, so it's making the inside more positive, causing that depolarization. Now, what's unique though to the pacemaker potential compared to our neurons is depolarization activates voltage-gated calcium channels. And it's the movement of calcium. So now in this depolarization, it's calcium coming in that further depolarizes it. Then whenever we reach that threshold, again, lots of calcium channels open, and the movement of calcium causes a huge depolarization. Again, different than the other cardiac muscle cells where we're talking about the movement of sodium that causes the depolarization. So repolarization occurs as in other ones. So when the voltage-gated calcium channels close and the potassium channels open. So calcium stops here at the top, sodium open, and now sodium's going out of our cells. But you can see this repolarization takes a little longer than a normal action potential that we'd seen with neurons. Then ultimately, as that resting membrane potential is reestablished and we're ready for another action potential to occur. All right, now the action potential in other cardiac muscles. So again, we saw that depolarization phase, but we have that rapid depolarization is due to changes again in sodium, potassium, and calcium. But again, we're talking about mainly sodium here is the primary determinant, not the calcium that we saw with the pacemaker. So we open those voltage-gated sodium channels. That allows for this depolarization to occur where we're changing the membrane potential. Okay, then we get this early repolarization phase. So this looks a lot different. We see this long kind of depolarization phase before we get it back to that resting membrane potential. So this early repolarization occurs whenever we get changes in the permeability. So this is when the voltage-gated sodium and some of the voltage-gated calcium close, but only a small number of the potassium channels open. So it takes longer for those potassium channels to open, which means that it's longer till we get back to rest. So sodium ions continue to move, it slows, but then again, we have that slower movement of potassium. Okay, then we have this plateau phase here, and we kind of have a gap here in the time, but this plateau occurs as voltage-gated calcium channels remain open, and now we have the movement of calcium and some sodium into the cell, so it's still staying depolarized. So again, the slow movement of potassium out of the cell, as well as voltage-gated calcium staying open, causes this long plateau phase. Then we do get that final repolarization. So final begins whenever those calcium channels close, many more of the voltage-gated potassium open. So now calcium stops coming in, sodium can move out, and we get that resting membrane potential restored. So the differences then between skeletal and cardiac muscle. So again, with cardiac muscle, those action potentials are conducted from cell to cell. In skeletal, the action potentials go along the length of a single fiber. In cardiac, the rate of action potential is slow because, again, we have those gap junctions and a small diameter of fibers, whereas skeletal action potentials are very quick due to that larger diameter. In cardiac, we have that prolonged period. So again, that plateau phase is unique to cardiac muscles. Skeletal muscles, there's a rapid repolarization, so there's no plateau. And then also in cardiac, the calcium-induced calcium release, the movement again of calcium through the membrane and T-tubules release calcium from the sarcoplasmic reticulum, further increases the amount of calcium that maintains that depolarization. So skeletal muscle doesn't depend on this mechanism. It relies on intracellular calcium only for that contraction. So this is kind of a visual here to compare these. So again, in skeletal muscle, we get the depolarization. So that's really the same. In cardiac and skeletal, we reach the threshold, we get depolarization. But again, with skeletal muscle, repolarization happens very quickly. So it rapidly goes back as potassium channels open and voltage-gated sodiums close. But in cardiac muscle, those sodium and calcium channels stay open, calcium remains inside, and the potassium slowly opens. So we get this long plateau phase that's unique to cardiac muscles. So there are still some refractory periods. So absolute, refractory. Again, this is where it's completely insensitive. So it doesn't matter that you keep stimulating it. So in that plateau phase, you can keep trying to depolarize it, but it's already depolarized. So that's in a refractory, you can't stimulate it again. And then that relative examine, it's reduced sensitivity. So it could if there was a big enough stimulus, but it's unlikely. So these are longer refractory periods due to that plateau phase, but it prevents tetanic contraction, so more like spasms. We don't want to have long, like spasmic activity like tetany, but it allows for more controlled contraction. And then finally here, looking at an electrocardiogram, this is what's, looking at that electrical events of the myocardium, again, that middle wall can be correlated with these mechanical events. So when we're looking at an ECG, we have a few different waves that we look at. So the P wave that's shown here, that's the atrial depolarization. And that signals the onset of the atrial contraction. So that's going to happen first. Again, the atria contract rather quickly. Then we have this QRS complex that's here at the peak, which is the ventricular depolarization. That signals the onset of the contraction of the ventricles, and then we get repolarization of the atria happening kind of at the same time. So we get contraction, depolarization of the ventricles while the atria are repolarizing. Then we have this T wave, which is the repolarization of the ventricles that precedes the relaxation. So it remains contracted during this time of this ventricular repolarization. And you can see that it's longer than what our atrial depolarization is. So then we have this PQ interval. So this is, or just PR interval. So it's about 0.16 seconds. And this is when the atria contract and begin to relax and the ventricles begin to contract. So that's that time that it takes from the atria contracting to the ventricles contracting. It's called the PQ or PR interval. And then finally the QT interval. So this is when the ventricles contract and begin to relax. So how long those ventricles remain contracted is the QT. How long the atria are contracted is the PQ or PR.